Hello students, today we will continue with the fermentation process. In the previous lectures we have studied about uh, respiration, introductory part, difference between the aerobic and anaerobic respiration, and the common pathway in both type of respiration, that is glycolysis. The last lecture we studied about the all the steps of the glycolysis. So after that, uh, glycolysis starts from the glucose as a respiratory substrate and ends at pyruvic acid, which contains three carbon. So in the end, we get two molecules of pyruvic acid, and the process of glycolysis was taking place in the cytoplasmic part of the cell. After that, here we will discuss about anaerobic pathway, means the pyruvic acid will undergo fermentation if oxygen is absent. So what is fermentation? There are three major ways in which different cells handle the pyruvic acid produced by the glycolysis. One is lactic acid cycle, lactic acid fermentation, you can say, alcoholic fermentation, and aerobic respiration. So the upper two, lactic and alcoholic fermentation, is the method of anaerobic respiration, and the third one is aerobic respiration. Okay. So, lactic acid fermentation first. Uh, well, in the lactic acid fermentation, the pyruvic acid is converted into lactic acid. It takes place in the muscles in the anaerobic conditions. When this condition appears during vigorous contraction in the muscles or during the vigorous exercise, then our muscles do not get sufficient oxygen supply, due to which the glucose which is present there will converted into lactic acid through lactic acid fermentation. But later, when it gets sufficient amount of oxygen, it finally completely undergo breakdown to produce CO2, water, and large amount of ATP. But up to that condition, when anaerobic condition is there, it converted into lactic acid, and lactic acid creates pain in the muscle. Okay, so the reaction catalyzed by the lactate dehydrogenase, means which enzyme is utilized for the lactic acid fermentation? It is lactate dehydrogenase. Lactate dehydrogenase. NADH plus H plus is reoxidized into NAD plus, means here a loss of NADH plus H plus is taking place. Means you have seen in the glycolysis part when one molecule of NADH plus H plus was formed in the sixth step, but here it is reoxidized into NAD plus, so it is a loss. Okay, so you can see here this one is the glycolysis process. In short, it is given here up to the pyruvic acid, and this pyruvic acid now uh, this pyruvic acid, it will enter into the lactic acid fermentation. This is the lactic acid fermentation. So with the help of an enzyme, that is lactate dehydrogenase, this pyruvic acid will convert into lactic acid. And during this process, the NADH plus H plus is oxidized to form NAD plus. This NAD plus was formed this sixth step of the glycolysis. So here two molecules were formed because two molecules of glyceride 3-phosphate was converted to 3-phosphoglyceric acid. In similar way, there are two pyruvic acids. So two pyruvic acid will convert into two molecule of lactic acid. Therefore, all these are two in number. Two NADH plus H plus were formed initially, and here two NAD plus is formed in the lactic acid fermentation. Okay, so NAD plus NADH plus H plus net effect is 0. So overall, if you see, only two ATP is formed, which is formed during the glycolysis process. Okay, so in lactic fermentation, only two ATP is produced. Okay. Okay. Now the alcoholic fermentation, means it occurs in two steps. In the last part, you can see this one is the alcoholic fermentation, and it is two-step process. Pyric acid finally converted into ethanol plus carbon dioxide, and in this process also NADH plus H plus is oxidized to NAD plus. So the net energy through NADH plus H plus is zero here. Okay, and only two ATPs again formed, which already formed during the glycolysis process. So in the fermentation process, we do not get much amount of energy like in the aerobic respiration. Okay, here only two molecules of ATP net we get. Okay, so here you can see all the points. It is a type of incomplete oxidation. Alcoholic fermentation, a type of incomplete oxidation of the glucose, and sets of reactions where pyruvic acid is converted into CO2 plus ethanol. Enzyme pyruvic acid decarboxylase is utilized in the first step, due to which carbon dioxide is removed. Okay, and in the second step, alcohol dehydrogenase enzyme is utilized, due to which ethanol is formed. Okay, so in this way, in alcoholic fermentation, two-step reaction is there. In the first step, pyruvic acid release one molecule of CO2 through the enzyme pyruvic acid dehydrogenase, and in the second step, it forms the ethanol when we utilize alcohol dehydrogenase enzyme. Here, NADH plus H plus is oxidized in the second step into NAD plus. So energy released is less than 7% of the energy in the glucose. Means all the energy which is present in the glucose, out of that less than 7% energy is obtained in this reaction only. Okay, less than 7% we get. And alcoholic fermentation, where it occurs, yeast poisons to death when concentration of alcohol reaches about 13%. It means to say, during alcohol fermentation is taking place in the yeast cells, but yeast can never produce the alcohol more than 13%, because alcohol, excess amount of alcohol can cause the death of the cells, because it creates dehydration. So yeast cell will itself, it will remove all its water content. So yeast can never produce the alcohol concentration beyond the 13% limit. Okay, so when it reaches up to the 13%, alcoholic fermentation stops, and after that, when this percentage is reduced, then again fermentation starts. So if we get the alcohol, ethanol, more than 13%, then there is need of distillation. Okay, while the beer and wine, which is undistilled product, means it is directly it is obtained from the yeast fermentation, so it contain the alcohol concentration less than 13%. Okay, next is aerobic mode of respiration. So aerobic is the complete oxidation. It is complete oxidation of the glucose. Energy extracted, synthesized in the form of ATP, means large amount of ATP is obtained, common in higher organism, and it is taking place within the mitochondria. The fermentation process, which we have just studied, it is taking place in the cytoplasm. But the aerobic respiration, after the glycolysis, the part is taking place inside the mitochondria. In this process, oxygen is required, while for the fermentation, oxygen is not required. For the glycolysis, also oxygen is not required. Okay, and in this process, CO2, water, and large amount of energy will be obtained. Aerobic respiration. The pyruvic acid will enter into the mitochondria. So, for transfer of this pyruvic acid into the mitochondria, there is some reaction also. So, two main event of the process, which the detail is we will discuss here. Um, but initially, here in short, it is explained here what is taking place actually. The complete oxidation of the pyruvate by the stepwise removal of all the hydrogen atoms, leaving the three molecules of CO2 matrix of the in the matrix of the mitochondria. Okay, so what are the things which is taking place in the matrix of the mitochondria? For their complete oxidation of the pyruvic acid will be taking place in the stepwise reactions, due to which all the hydrogen atoms will be removed and 3 CO2 molecules will be released. And the second main event, the passing on the electrons removed as part of the hydrogen atoms. The passing on of the electrons removed as a part of hydrogen atoms to the molecular oxygen with simultaneous synthesis of the ATP at the inner mitochondrial membrane. So in these two very short para, all the what are the things which will taking place in the main reaction is given here. So in the first main event, and the first main event will taking place in the matrix of the mitochondria, while the second main event will taking place at the inner membrane of the mitochondria. In the first step, hydrogen atoms it will remove one by one, along with the 3 molecules of CO2 will be removed, while in the second main events what will take place, electrons will removed from the hydrogens and molecular oxygen will be there, which will convert into water. Okay, this the hydrogen, electrons and oxygens, these are the some combinations which will produce the water, and this reaction will taking place in the inner mitochondrial membrane. Okay, so here a short reaction is taking place before these two main event, because this time pyruvic acid is present in the cytoplasm, then it will enter into the mitochondria, and this main event is known as Krebs cycle. Second main event is known as electron transport chain. So this Krebs cycle and pyruvic acid means, which is the product of glycolysis, there is a link reaction will be taking place to connect these two. What is this link reaction? This is given, link reaction is given here. Okay, so the pyruvate enters from cytosol, means from the cytoplasm, to the mitochondrial matrix and undergoes oxidative decarboxylation. Decarboxylation means removal of carbon dioxide. So what uh, enzyme will be utilized? Dehydrogenase and for the decarboxylase enzyme should be used, but here it is what enzyme is given, pyruvate dehydrogenase coenzyme A, NAD plus. Okay, this is actually a complex enzyme which will perform three different functions. Pyruvate dehydrogenase is the complex enzyme which performs three different functions. One is decarboxylation, means removal of CO2. Second one is removal of hydrogen, that is dehydrogenase. And third one is the acetylation. Acetylation means addition of acetyl group. Okay, you can see here, this is the pyruvic acid which is coming from the cytosol or the cytoplasm. Now, and it will enter into the mitochondria. Now here coenzyme A and NAD plus, these two are the substrate, and it will convert it into acetyl coenzyme A, means this pyruvic acid undergo acetylation, and this coenzyme A will add with the pyruvic acid, so it will convert it into acetyl coenzyme A. It is the product. Along with that we get CO2 which is released from the pyruvic acid, and the hydrogen which is released from the pyruvic acid will occupied by the NAD plus to form NADH plus H plus. Okay, so in this way, from the pyruvic acid, which is 3 carbon compound, it removes the CO2. So in the acetyl coenzyme only 2 carbons are present. Okay, it is 2 carbonic compound, and hydrogen is also released from the pyruvic acid. So pyruvic acid is 3 carbon compound, while acetyl coenzyme is 2 carbon compound. And as you know, there are 2 pyruvic acid were formed. In case of 2 pyruvic acid was were formed in the glycolysis. So here it is only one. It is given for one molecule, but this reaction occurs twice. This reaction occurs twice. Okay. So this acetyl coenzyme A now enter into the Krebs cycle. Krebs cycle is also known as TCA cycle or the tricarboxylic acid cycle, because the first stable product of this cycle is TCA, tricarboxylic acid. or you can say 3 carboxylic acid is present in the first product of the glycolysis, sorry, in the first product of Kreb cycle, therefore it is known as TCA cycle. So here in short some reactions are given. Condensation of acetyl group with the oxaloacetic acid and water to form the citric acid. This citric acid contain 3 carboxylic groups, okay, and it forms due to citrate synthase. So you can say this is pyruvate 3 carbon compound, which is the product of glycolysis. After that, link reaction is just we have read, which forms the acetyl coenzyme A, which contain 2 carbons. Now this acetyl coenzyme A enter into the TCA cycle. It is also known as citric acid cycle because citric acid is the first product of this cycle. It is also known as tricarboxylic acid cycle because the citric acid contains 3 carboxylic acids. It is also known as Kreb cycle because it was discovered by the scientist Kreb. Okay, so what happens? Acetyl coenzyme A combined with the oxaloacetic acid, okay, and water to produce the 6 carbon compound. This is acetyl coenzyme A contain 2 carbons, oxaloacetic acid contain 4 carbons, and both will combine along with the water and with the help of enzyme citrate synthase to produce citric acid. Okay, now the sequence of reactions, again it is 10-step reaction, and ultimately oxaloacetic acid will reform, and this cycle also occurs twice the time because total 2 pyruvic acid we get from one glucose molecule in the glycolysis, but it is given for only 1 pyruvic acid, so this reaction will taking place 2 times. Okay, so in the sequence after the citric acid, CO2 at some position CO2 will remove, NADH plus H plus will form because hydrogen is also removed, and it will when CO2 is removed, so 5 carbon compound will be obtained, means alpha ketoglutaric acid. After some time again CO2 is removed and one hydrogen is removed to produce NADH plus H plus, and the again the 7th step here, and one phosphate will remove, which is obtained by the ADP to produce ADP, and in this sequence succinic acid will be formed which contain the 4 carbon compound. Now the FAD at some position after the succinic acid, again hydrogen is released. This hydrogen is taking place by the FAD plus to produce FADH2. This is also a form of energy assimilatory power like NADH plus H plus. Okay, FADH2 is also assimilatory power, and then finally it is converted into malic acid, and again in a sequence hydrogen is released which NAD plus will get the hydrogen to produce NADH plus H plus, and ultimately oxalosuccinic acid is, sorry, oxaloacetic acid OAA will reform. Okay, so it is a cycle, so oxaloacetic acid is reformed, but during this reaction we get CO2 1 2 at 2 positions, we get NADH plus H plus at 3 positions 1 2 and 3, we get ATP at 1 position and FADH2 at 1 position at 1 times. Okay, so 1 ATP molecule, 1 FADH2 molecules, and 3 NADH will be get, and 2 CO2 molecule will be obtained. Okay, in the Kreb cycle. So this is for the one cycle. When the cycle repeats for the second pyruvic acid, then again we will get the same thing. Okay, means 1 FADH2, 3 NADH2, 2 CO2. Okay, now this is about this reaction. So this is in single step what we get, it is given here. After the pyruvic acid we utilize 4 molecule of NAD plus, 1 molecule of FAD plus, 2 molecule of water, which is also given in the Kreb cycle, 1 molecule of ADP, and 1 inorganic phosphate is also utilized, and in the mitochondrial matrix this reaction is taking place and various enzymes are there, and ultimately we get 3 CO2 molecule, 4 NADH plus H plus, or you can say 4AH plus OH plus. So in the Kreb cycle we get 3, and in the link reaction 1 NADH plus H plus is obtained in the link reaction, so total 4, and CO2 is also 2 in number in Kreb cycle, 1 from the link reaction, and 1 FADH2 in the Kreb cycle, and 1 ATP molecule is also produced in the Kreb cycle. So this is the overall product in the Kreb cycle plus link reaction. Okay, so this is for 1 pyruvic acid molecule, but as you know that there are two molecules of pyruvic acid formed up, in the process of glycolysis, so this reaction repeats itself. So you can multiply all the things by 2 to get to know the actual products and to know the exact amount of product. Okay, so during the whole process of the oxidation of the glucose produce CO2, 10 NADH2, 2 FADH2, 2 ATP or GTP may be there in some cells. Okay, you can see this is 1 ATP, but we get 2 ATP for the second pyruvic acid. 1 FADH2 we can see from the one pyruvic acid, from the second pyruvic acid again we will get the same thing. And 4 NADH2, 4 NADH2 in the link plus Kreb cycle, 4 to the 8 for the second pyruvic acid, so 8 NADH2, but 2 2 we get from the glycolysis. In the sixth step of the glycolysis, 2 NADH2 were produced there, total 10 NADH2. Okay, and total 6 CO2 molecules is also there. 6 molecule, how it is 6? You can see 3 CO2 from the one pyruvic acid and multiply by 2, total 6 CO2 will be obtained. Understood? Now the second main event is the electron transport system, and one more reaction is taking place in the end, that is known as oxidative phosphorylation. So electron transport system and oxidative phosphorylation is given. This I will discuss in the next lecture. It is also simple process, not a complex. You can see here all the hydrogens which we get here, all the hydrogens, all the electrons, means the same number of electron also. The hydrogen amount, the as much as hydrogen atoms is produced, in the same number electrons are also produced. So these electrons will undergo electron transport chain. How it will get, we will discuss later. And when this electron will pass through electron transport chain, in the end it will produce a water molecule, plus due to this electron transport through the various proteins is also involved in the ATP production. This ATP production is known as oxidative phosphorylation, because phosphorylation means addition of phosphate. ATP will be formed, and because this electron is transferred due to the oxidation process or respiration process, therefore it is known as oxidative phosphorylation. So this is a mixed type of reaction. Therefore it is known as ETS for the electron transport and oxidative phosphorylation for the ATP production. How it will taking place, this we will discuss in the next lecture. Thank you, class.